I'm going to go over some chemical control methods, but I do not need you to memorize anything about any of them. What I really want you to understand for the sake of the exam is, so in an exam, I will give you a chemical control method and I will tell you what it does. I'll say it targets this anatomical feature, or it targets that anatomical feature, or it targets this kind of molecule or that kind of molecule. Your job then is to figure out which kinds of those hard to kill microbes would be most resistant or most vulnerable to the chemical control method. So you don't have to remember what phenol does, you don't need to remember what quats do. I will tell you in the question what kinds of molecules or anatomical features they target, and then you have to take what I told you about the chemical control method and use what you've remembered, what you've studied about those hard to kill microbes, and figure out which hard to kill microbes it would kill. Okay. So the efficacy of some germicidal chemicals can be altered by the presence of organic matter, like dirt, for example, or grease, and can also be altered by pH because it can change the structure of the chemical. Care should be used to use the correct concentration as well, and to adjust concentration if organic matter like cotton balls are involved. So it turns out soaking cotton balls in, for example, quats, which is a type of chemical biocide, makes the quat less effective and more likely to harbor resistant bacterial strains, which can lead to infection when the cotton balls are used to sterilize wounds. So there is a story about this hospital that was pre-soaking cotton balls in quats and using those cotton balls to swab and to clean wounds and things like that, or to prep a site for a shot. But the problem is, because the organic material was making that biocide less effective, a quat-resistant bacterial community started living in the cotton balls that had been soaking for days, and those were getting spread around to different patients. So like I said, we're not going to go into the different types of chemical controls in depth, but we will talk more generally about how they work, and we'll also talk about how to test their efficacy. So let's talk about the disc diffusion test right now. Disc diffusion, which can be used to test efficacy of antibiotic drugs as well as biocides, is shown here. So this is sort of how this works. What you do is you inoculate a petri dish that has media in it with bacteria. So the media is the gelatinous stuff that's filled with nutrients for the bacteria to grow. So you take this petri dish and you inoculate it with one species of bacteria, and then you place these discs, these paper discs, that have disinfectant on them, on the plates. And then you let the bacteria grow for a while, and then you examine what are called the kill zones, or the zone of inhibition. Now you can only test one species of bacteria at a time here. Each dish is inoculated with a different bacterial species. So you have Staphylococcus aureus, which causes staph infection, right here. And this is a gram-positive bacteria. And this lighter yellow, that's the bacterial film that's growing. And then this darker yellow, that's where the bacteria is not growing. That's seeing through just to the media behind. And then on on this dish, you have E. coli, Escherichia coli. This is a gram-negative bacteria. And then you have Pseudomonas aeruginosa, which is not only a gram-negative bacteria, but it's that extra hard-to-kill bacteria. Remember, we talked about in the hard-to-kill microbes, we talked about Pseudomonas and Burkholderia, so they have those extra bells and whistles that make them even harder to kill than most gram-negative bacteria. So then you have these paper discs, and each dish has the same 4 paper discs. You have these are various disinfectants or germicidal agents. You have chlorine, a phenol, which is a kind of phenol. You have quat. You have hexachlorophene. So these are all different disinfectant chemicals, and they're all the same on each of these dishes. But you can see that they work differently for different species of bacteria. Notice that everything works the best on gram-positive bacteria, right? And then they work worse on the 2 gram-negative bacteria species. And remember, Pseudomonas is a special case of gram-negative with pores that expel chemicals, so this is the most resistant to everything. Actually, interestingly, it looks like maybe chlorine might work. It's hard to tell, but it might work it works out equally or maybe even a little bit better on gram-negative than gram-positive, right, for at least this species. All right, so unifying features of chemical control methods. These are chemicals that damage critical anatomical features on a microbial species. Different chemicals may damage different molecular structures based on the chemical's chemistry. The most effective chemical control methods tend to also be harmful to animal hosts, and so they're not for consumption. They're going to be disinfectants for inanimate objects or antiseptics that you put on the outside of your body but that you don't ingest. So here's some general mechanisms by which chemical control methods work to control microbial growth. You can dissolve the lipids. So anything that dissolves, any chemical that dissolves lipids is going to dissolve the cell membrane or any waxy structures, like for example mycolic acid on mycobacteria. There are chemicals that disrupt hydrogen bonds. Well, hydrogen bonds are essential for the structure of DNA, the structure of RNA, for protein structure, and so it's going to cause denaturation of all these molecules, which are essential for life. A chemical that disrupts disulfide bridges. Well, disulfide bridges are really important for holding the three-dimensional shape of enzymes together, so that's going to cause protein denaturation, or so the three-dimensional structure of any protein, not just enzymes. And then some chemicals can induce covalent bonds between amino acids that wouldn't normally form covalent bonds. So that's going to change the three-dimensional structure of proteins as well. So that's going to be protein denaturation. And again, if you don't know at this point the difference in chemical structure between protein, nucleic acid, lipids, sugars, carbohydrates, you need to review that from your prereqs. I try to go through a lot of prereq material in this class, but I can't go through all of it. You do have a chemistry chapter in your textbook that you could use to go back and remind yourself, what are proteins, what are nucleic acids, what are lipids, what are carbohydrates. We've covered it a little bit, but if it's too rusty, you should, we're getting into a little bit of chemistry here, you should probably review that material. All right, phenolics. So here is a phenol molecule. All phenolics have at least one of these groups, but other parts may be included. So in chemistry, especially in organic chemistry, there's so many carbons that people get, chemists get tired of drawing them, biologists get tired of drawing them. Every corner where you don't see the element labeled, just assume it's a C. This is a carbon. And there's alternating double bonds between carbons and single bonds between carbons. So this phenol ring, you'll see that at least one of those on any phenol molecule, although it might have other sections as well. These are reactive because of the hydroxyl group and also because of these double bonds between carbons. Both of them are very reactive, so they're going to force chemical reactions with other molecules that should be stable in the cell. Right, so those double lines indicate that two pairs of electrons are shared between the carbons, but one of those pairs is capable of forming bonds with other molecules, so you could take this line and reform it with another molecule that might have been essential to the cell, and that would disrupt the other molecule. These mess primarily with the plasma membrane, but they can react with other cell parts as well. It's not only the lipid bilayer that it affects, that's just something that's the most vulnerable to it. And so these are sterilizing agents because everything has a phospholipid bilayer. I mean, except for viruses, right? So they have some action on other molecules, but it'll take them longer to disrupt other molecules, and they disrupt lipids right away. Halogens. So halogens are any element in this column of the periodic table. They have only one electron missing from their outer shell. So there's fluorine, chlorine, bromine, iodine, right? Chlorine and iodine are probably the most familiar to you. And chlorine and fluorine and iodine probably are pretty familiar. So these mess up hydrogen bonding between molecules, often by stealing hydrogen atoms. Hydrogen bonding we discussed when we talked about water, right? So that's, attractions between molecules without sharing electrons. The hydrogen bonds are also important in maintaining the three-dimensional structure of enzymes, in holding two strands of DNA together, and the function of RNA involved in translation, like transfer RNA, ribosomal RNA. So all of those kinds of molecules that are involved in the genetic code and converting it to protein products, that's going to disrupt that whole thing. Gene expression from DNA to protein is going to be disrupted by these. So these are sterilizing agents as well. All right, alcohols. These are disinfectants. They don't work against endospores or non-enveloped viruses. They work by dissolving the plasma membrane in oils. So alcohol is an amphipathic molecule as well. It can dissolve in water and also in lipids, because this section, so this is ethanol. Ethanol has 2 carbons. This is a hydrocarbon section. It's very tiny, but it's still a hydrocarbon. So this piece is hydrophobic. This piece is hydrophilic. And so they can, because of the hydrophobic region and the hydrophilic region, what they do is they can blend with the hydrophobic portion of the plasma membrane or other fats and oils, but then also allow water to wash them away, sort of like a soap. They also cause proteins to coagulate or clump up together. These are not good for wound cleaning. They coagulate proteins at the surface of the wound, and that allows bacteria to live beneath. Heavy metals, this could be like mercury, silver, copper, zinc. So we use these today for halting microbial growth. So mercury is a preservative, especially in, it's not used in vaccines anymore except in the flu vaccine, but you can get it without. But that was a place where we used it a lot as preservative in vaccines. Silver, silver used to be used by the Romans in water barrels. They'd put silver coins in water barrels to halt microbial growth. We still use it in newborns' eyes. So when a newborn comes out, sometimes you can get them silver drops to prevent eye infections. We also use silver to clean catheters. Copper you find in anti-mildew agents. Zinc you find in hand sanitizers and mouthwashes. So these are all examples of heavy metals. And what they do is they affect sulfydryl groups in proteins, which causes protein denaturation. So let's just say these are strings of amino acids. So it's the carboxyl amino group, carboxyl amino, carboxyl amino, carboxyl amino, and it goes down, down, and folds back on itself. So the green line is the repeating amino acid chain that a protein is made out of, and it's folded back on itself, and these folds are being held in place by a covalent bond between a sulfhydryl group of this amino acid and the sulfhydryl group of this amino acid on a different part of the chain. And so that's holding this shape together. Well, this heavy metals disrupt these sulfhydryl groups, and that's going to cause the protein shape to unfold. It's not going to, if these sulfhydryl groups are disrupted, they're no longer going to be holding these pieces of the enzyme together, and so the enzyme will denature. So this is biocidal. It's not, there's a lot of, you're not going to kill everything. So it depends on what microbe you're talking about, how much you're using and stuff. So it does slow microbial growth or halt it, or maybe it sterilizes some species of completely removes some species of microbe. But you can't really use these as a sterilizing agent. Quats. Quats are antiseptic. The word stands for quaternary ammonia compound. We're not sure, the mode of microbial action is still unclear. Based on the kinds of microbes that are affected by quats, we think that it disrupts the plasma membrane specifically, but not all lipids, because it doesn't work against mycobacteria. It doesn't work against the mycobacterial mycolic acid cell wall. So it doesn't disrupt all lipids, but it disrupts the plasma membrane, I think, or we think at least, researchers think. So it works on enveloped viruses specifically, so the viruses that have a lipid envelope, which is the cell membrane taken from the virus that it came from, taken from the cell it burst out of. It works on against most bacteria, it works against protists, it works against fungi. So those are all things that have this lipid and a lipid cell membrane or lipid envelope. It does not work on tuberculosis, so the mycolic acid cell wall is protective. Does not work on non-enveloped viruses which have no lipid envelope. Does not work against endospores, which have a protein case protecting their lipid bilayer. Aldehydes, this is going to be stuff like formaldehyde, and this is an aldehyde group, so aldehydes all have this group somewhere on it. These are disinfectants. They form cross-links between several R groups on proteins. This causes enzymes to degrade or denature because it links up amino acids that weren't supposed to be linked in the protein, which changes its shape. Reactive oxygen species, which you learned about already. So examples would be hydrogen peroxide. So if you add reactive oxygen species to something, even if it's an aerobic, so all anaerobic organisms are going to be negatively affected by reactive oxygen species because they can't even take the ambient levels. They don't have those enzymes to neutralize reactive oxygen species. But even if you're working with an aerobic organism that's infecting a wound or something, if you overwhelm their enzymes that neutralize reactive oxygen species, you can add more reactive oxygen species than the microbe can handle, than the aerobic microbe can handle, right? So you overwhelm their catalase, their superoxide dismutase, their peroxidase. So what this does is it creates free radicals in the cell. Free radicals are molecules with unpaired electrons, and they just wreak havoc on all the essential molecules of the cell. Reactive oxygen species can be antiseptics or sterilizers. And remember, ionizing radiation creates reactive oxygen species inside the cell, and so that by interacting with water, and so that's the main mode of action for ionizing radiation. But you can also just dump hydrogen peroxide on stuff. Right. Food preservatives. Well, this is just a very broad category. There's lots of different kinds. But for the molecules we just finished discussing, you don't have to worry about it being safe for ingestion so much, right? Because you're not ingesting them. You're sterilizing something in a outside of the body, or at least on the body's surface. Food preservative has to be safe enough to ingest at least in small quantities. So examples would be nitrates. Nitrites inhibit the production of iron sulfur clusters, which are essential for botulism metabolism. So you see how that's really specific? It's like you're just preventing botulism from growing in the food when you're using a nitrite. Okay. So different food preservatives will have different modes of action. They'll have different levels of toxicity. So they're hard to talk about as a group, but that's something to remember. It's out there as a chemical control method. Of course, antibiotics, there's lots of different kinds with different modes of action. Again, they have to really target the microbe and not attack eukaryotic human cells, right? No harmful action against eukaryotic cells. Have to be safe for humans. So trying to kill a fungi or a protist, it has to be active against eukaryotic. So that's another thing. An antibiotic, you can have an antifungal. So when doctors say antibiotic, they mean antibacterial. An antibiotic means an anti-life, but they mean antibacterial. So when you hear antibiotic, people are usually talking about antibacterials. But there's also antifungals. So if you're trying to kill a fungus or a protist, those are eukaryotic, right? So then you have to be even more careful. You have to find something that's active against eukaryotic cells that are not animal cells. Then we have plasma and supercritical fluids, and these are both sterilizers. Plasma is they're high energy molecules so these are, well, I should just say both of these are very high energy molecules that cause damage to other molecules. Plasma specifically is gas particles that are so excited that the electrons have separated from the protons in the nuclei of the atoms. So you have many positive charges that have separated from all these negative charges, and they readily interact with all other molecules. They steal electrons, add electrons, changing bonds everywhere. They wreak havoc on any molecule they touch. Supercritical fluids are substances that are kept on the edge of a gas and liquid phase. They are very, very good solvents. So these are really good sterilizing techniques, but very expensive. They're used in hospitals. Supercritical fluids are gases that have been compressed with pressure down to a fluid, but they might be at normal temperature. So they can be used to sterilize tissue transplants, which you couldn't, put into an autoclave or something at high heat. Plasma is great for sterilizing things like tubing that is difficult to wash. So to give you an idea of so here, this is a table that's, I think it's not in the textbook. This is from a textbook that predates the one you're currently using, but I still thought it was useful. So this is just a quick table of these various chemical controls and whether they kill endospores or mycobacteria, which are two of the hardest bacterial structures or bacterial forms to kill. So this is a a phylogenetic group of bacteria. Some bacteria form endospores and others don't. But endospores are very hard to kill, and that mycolic acid is really hard to penetrate in mycobacteria. So these are some of the things that doctors tend to worry about the most because they're the hardest to kill. So this chart gives you some of the chemical controls that we covered and tells you what they do and don't kill. You can take a look at this and see if you can figure out why, because that's really what I'm going to want, is based on what we've discussed, based on what I tell you about what anatomical features these chemical controls target, what microbes can they kill and what can't they. And I will give you a very specific example here, okay? So if I gave you a test question about chemical controls, this is why I'm saying like that whole lecture on chemical controls were just a list of examples you don't need to memorize. I want you to memorize anatomical features of hard to kill microbes. That's what I want you to memorize. Because you don't need to memorize the chemical control methods, because I will just tell you. There's many, many other chemical control methods out there that we haven't covered. So I want you to figure out, I want to teach you how to think about it, not how to memorize all of them. Okay? Because you can just Google search any of them in real life, right? So this is kind of what I'd ask you on a test. If I wanted to ask you about phenols, I would tell you what phenols do. I'd say phenols dissolve and disrupt lipids. They target lipids more than anything else. Now, keeping the anatomy of endospores and mycobacteria in mind, why would phenols be effective against mycobacteria but not endospores? Okay. So listen again. Phenols dissolve and disrupt lipids. They target lipids. Those are hydrocarbons, right? That's going to be like fats, waxes, things like that, steroids. Okay, keeping the anatomy of endospores and mycobacteria in mind, why would phenols be effective against mycobacteria but not endospores? Now, to approach this question, what you have to consider is, what is the protective layer for these different kinds of microbes? So you have mycobacteria and you have endospores. What is the protective layer? Now, endospores and mycobacteria both have plasma membranes that are made out of a phospholipid bilayer. So of course, if a phenol got into the cell past the protective barriers, it would kill both mycobacteria and endospores. But both mycobacteria and endospores have a shield to prevent chemicals from getting into the cell membrane. So before the phenol can act on the cell membrane, it has to penetrate this outer protective layer. What is that protective layer for mycobacteria and endospores? And how would phenol penetrate one versus the other? Well, here's mycobacteria. Remember, mycobacteria have this peptidoglycan cell wall. Remember, the cell wall is not a chemical shield, but the mycolic acids are a chemical shield. And the mycolic acids are a wax, which is a lipid. So this explains, right, why mycobacteria would be sensitive to phenols. Their protective layer is made of lipids, right? It's a solid lipid, it's still a lipid. The shield for mycobacteria is a lipid. So you douse it with a chemical that destroys lipids, you destroy its shield. And now the phenol can get into the cell, where the cell membrane is, and destroy that as well. Endospores, on the other hand, their final outer layer is a protein coat. So where is that part here? Spore coat begins to form around the forespore. And then the forespore is exocytosed from the mother shell. So this outermost layer for endospores is a protein coat. Endospore layers go, protein coat is the outermost shield. Then you have two phospholipid bilayers, and between them is a peptidoglycan cell wall. So you have protein coat, phospholipid bilayer, cell wall, phospholipid bilayer. Now, remember, cell walls are not shields. And yes, the phospholipid bilayer is sensitive to phenols, because phenols will disrupt lipids. But if the phenol can't even get through, the problem is the phenol's not going to get through that protein coat, right? The protein coat is going to be a shield against phenols. Now, if you soak an endospore in phenol long enough, the phenol will start to act against the protein coat, but the phenols act most effectively against lipids. So this endospore is going to be the protective shield of the endospore is protein. Phenols aren't that effective against proteins. The protective shield for mycobacteria is a lipid. Phenols are active against lipids. Phenol is going to destroy this shield for mycobacteria before it destroys this shield for endospores because it acts against lipids more than it acts against proteins.